We do quality control from the biofermenters until the final products. So we're gonna count the viable cells, the colony forming units per milliliter of liquid inoculant, and we're gonna count it also in the final product. So if you have the granules or the powders with the rhizobium on it, we're gonna do a method to extract the bacteria and count it per gram of product. So we do the quality control from the beginning until the end of the production and formulation process. We also check the quality on plants. So we're gonna do very small bioassays on plants just to make sure that the roots will produce the nodules and will produce active nodules. So we're gonna check on each root system the number of nodules, the size of nodules, the distribution of the nodules, and we're gonna make sure that they are active. So we're gonna chop the nodules and look at the color inside, and it will indicate us if the bacteria are active or not. So it's part of the quality control that we run on the bacteria. About the quality control that we do on the mycorrhizae, we control the quality from the production facility where we get the spores, the pure aseptic spores, and then we're gonna check the quality of the spores during the formulation, after the formulation process, so on the granules, on the powder, on the soil substrate, and then we're gonna check the quality of the spores in vivo, so on plants. We're gonna, at the different steps, we're gonna count the spores, we're gonna make sure that they are viable, we're gonna check the quality, just make sure that the spore will be filled with the lipid reserve that they need to get in action when applied in the field. So we really look at the spore under microscope to make sure that the quality is there, that the spores are intact, because the production and the formulation will bring a lot of stress on the spores, so we have to make sure that they went through these steps correctly and that at the end they will be viable and of top quality to make the difference with the plant. So it's part of the quality control, but at the end we will also validate that these spores will make the symbiosis with the roots plant. So we're gonna check on plants in greenhouses that we're gonna have the intra-radical network produced by the fungi. So by having the intra-radical network, we know that there is also the extra-radical network, the hyphae, the mycelium in the rhizosphere that will do the job of feeding the plant and hydrating the plant. So we're gonna have different methods to check inside the roots. So we're gonna stain the roots and we're gonna detect all the mycorrhizae structures inside the root system. And so we can confirm that when we inoculated first these plants, the mycorrhizae spores were viable, but were top quality and able to create the symbiosis with the roots plants. So we check the quality from the beginning until the end on all the different formulated products, the granules, the powders, the liquid formulation. We're gonna count the spores, check viability, and check the efficiency of the spores. So it's very important to control the quality on our products.